Today we see the process of DNA cloning in bacteria using a plasmid vector. Suppose for DNA cloning we first use the target DNA which contain our gene of interest, and we isolate this target DNA from genomic library or cDNA library. Then we use a vector. What type of vector? Cloning vector. For example, plasmid cloning vector. Now this cloning vector is bind to your target DNA. When this cloning vector is binding to a target DNA with the help of the enzyme ligase, it is known as the recombinant DNA. There is a no binding occur. This is your non-recombinant DNA. Now we have to introduce this into specifically into a host known as E. coli. So when we introduce this into a host cell, E. coli, we see that there is a bacterial chromosome, and there is an insertion of your recombinant part, and there is an insertion of your non-recombinant part, and there is an insertion of nothing. So we get a two types of cell, transform cell and non-transform cell. In the transform cell, we see there is an entry of the part, but in the some part there is a recombinant entry and there is a non-recombinant entry, and the non-transform cell there is a nothing. So we have to remove this non-transform part and select the transform cell which contain your recombinant DNA. So we select only the transform cell from both of this. We say only that both are transform cell but we select only the transform cell that contain the recombinant part and amplify it, multiply it in a fermentation tank. This is all about the DNA cloning in bacteria by using a plasmid vector. Thank you for listening. Ciao, ciao.